Okay, welcome back. As we wind up, okay, so we were talking about the telomere, and we've talked about the structure, how, and also the shortening of the telomere, and the function of the telomere being to protect the genome from nucleolytic activities or degradation, and also to protect against unnecessary recombination and repair, and to protect against interchromosomal fusion. And therefore, this leads to a very important function of the telomere, which is the preserving of the information in our genome. And then we have what is known as the telomerase. So the telomerase activity, which is simply to extend the ability to extend telomeres, is present in germline cells and also in certain hematopoietic cells. So when we look at the somatic cells, these actually have low or undetectable levels of telomerase activity. And so we find that actually these cells will undergo progressive shortening with DNA replication. So I find that telomerases are actually reactivated in most cancer cells, and also in cells that are immortalized cells and cancer cells. We find that actually telomerases are actually reactivated in these cancer cells. So no wonder we find that actually cancer cells are rather described as being immortal, was the replication, or the division will be unchecked and regulated. So this is the telomeres. And then we have the transposons. We have the transposons. We have the transposons. We have the DNA transposons. These are able to relocate from one spot to another. They are able to move from one genomic location to another. They're able to move from one genomic location to another by a cut and paste mechanism. So if you remember, if you're using a computer, you cut a document. You're going to move from a certain location that you had saved it, and then when you paste it in another location, it's been moved to that location. So the transposons actually move or jump from one genomic location to another by a cut and paste mechanism. And these transposons are actually power forces of genetic change. They can actually be used to introduce a piece of foreign DNA into a genome. And we can actually use these DNA transposons, or these DNA transposons actually very useful in the process of analyzing the regulatory genome. It is important, or they are important in the study of embryonic development. They are important in the identification of genes and pathways that are implicated in diseases, or even the pathogenesis as well. And these transposons also contribute to gene therapy. So these are sections of DNA that are able to move from one location of the genome to another. So they are jumping, they can jump from one location to another. So that was the chromosomal theory of inheritance. So we've looked at the theory of inheritance, of which we see that the chromosomes are the carriers of Mendelian factors, and meiosis being the basis of separating the factors into the gametes. We've looked at the structure of the chromosome. We looked at the chromatin. We looked at the alleles. We looked at the centromere. We looked at the telomeres. We looked at the sister chromatids. We've looked at the homologous chromosomes. We've looked at the non-coding DNA. We've looked at the DNA transposons and other information with regards to the chromosomal theory of inheritance. Okay, then the other point on the diagram that was there, we had a solenoid fiber. So showing a solenoid fiber. This is simply a secondary chromatin structure which helps to package eukaryotic DNA into the nucleus. So the solenoid fiber helps package eukaryotic DNA into the nucleus. So also, in addition to that, on the homologous chromosomes, when you look at the homologous chromosomes in somatic cells, we see that that is composed of 23 pairs. Now, of those 23 pairs, we find that we have 22 pairs which are autosomes, and then we have one pair of 6 chromosomes. So no wonder we say that these will contain a total of 46 chromosomes, So 23 pairs, which means that a pair means there are 2. So multiplying by 2 gives 46. Now, of those 23 pairs, 22 are autosomes, and a pair of it is a pair of 6 chromosomes. So having said that, come to the end of the session, and the next topic that we'll be looking at is cell division.